Duquesne University in Pittsburgh, PA, and earned her Bachelor of Science and Master of Science in Biology from Youngstown State University of Ohio. Her current dissertation research in the laboratory of Dr. Nancy Trun is based on microbial bioremediation of abandoned coal mine drainage and passive remediation systems in southwestern PA. Anna's research has been supported by grants from the National Association of State Land Reclamationists, NASLR, and the Geological Society of America through the National Science Foundation. She is currently a fellow in the ASM Future Leaders Program of California University's Advanced Science Communication Program. Anna?
Hi, everybody. Nice to meet you in person. Thank you for the introduction. All right, so my name is Anna, for those of you who haven't met me, and I'm currently a fourth-year PhD student at Duquesne University in Pittsburgh, Pennsylvania, in the laboratory of Dr. Nancy Trun, and today I'm going to talk a little bit about the work that we do. So we look at the influence of microbes, particularly bacteria, in these passive remediation systems, and what can be good or bad about it. So a little background here, which I'm sure most of you guys are probably familiar with as reclamationists, but still. So coal mining in Pennsylvania began in the 1700s, and it was unregulated until 1945. So because of this, we have inherited these abandoned legacy mines that result in abandoned mine drainage, or AMD for short. We know of about 11,000 mines in Pennsylvania that are both acidic and circle neutral, and this negatively impacts about 5,000 kilometers of streams within the state. Unfortunately, the state has fortunately, the state has a $29.1 billion recreation industry, but because these streams are polluted by AMD, that negatively impacts it. And to remediate AMD in Pennsylvania alone is going to cost somewhere between $15 to $50 billion. But tiny little silver lining is there was recently a federal pledge of $16 billion to address legacy pollution, of which about $11 billion was just for abandoned mines in particular. So AMD results in watershed pollution, and this is due to the exposure of ores naturally in the ground to watered oxygen, resulting in the production of sulfuric acid, which will then solubilize metals into solution. And we can have both circle neutral, not that button, both circle neutral systems, where we have this sort of orange color, orange, pink, yellow, orange boy. And these are typically the results of deep mines that are passing over limestone beds. This is one of the systems we study. This is the low pass remediation system, and that's actually some of our students right there sampling. And we also study this voice pass remediation system, which has this almost pretty sort of blue color, but just not natural. And this is also in about 30 minutes from downtown Pittsburgh. It's actually inside of a park. There's a ski slope right next to it that people go to all the time. So this is a surface mine. There's a couple of inlets into it, and it's much more acidic than this one here. So we look in particular at iron and manganese contamination within these ponds. And when I'm talking about the iron stuff, I'm going to have it noted in this sort of orange color. When I'm talking about manganese stuff, I'll have it noted in this sort of purple color. So iron will contribute to the total acidity. It has this orange-yellow staining that can happen. It can be toxic if you're consuming it in large portions. There's also an association between the development of cancer and increased risk of infections if you're consuming a large amount of iron. As for manganese, we also have staining and corrosion. We can have impair of DNA repair mechanisms. So this can mean that there can be more mutations in your DNA, which also can potentially lead to more cancer. There's also toxicity that can occur if you're being exposed to large levels of manganese, and this can result in cognitive impairments, especially in children that are being exposed to this, as well as the development of Parkinson-like symptoms, which is this sort of uncontrolled shaking, and also neurological effects in general. So iron and manganese, I'm not going to go super into chemistry. I'm a biologist, not a chemist. But we have the iron oxidation shells here, which each one of the little blue dots are an electron. And depending on how many of these you have, it changes your oxidation state. So the most common oxidation states that these are found in are denoted by the boxes here. And I'm just going to briefly throw nitrogen in here because we're going to talk later about the coupling of iron and nitrogen for nitrogen-dependent iron oxidation. So passive remediation AMD is appealing because it's a lower cost and it's low maintenance. You basically set these systems up, let them go, and they kind of take care of themselves. And they're usually composed of these very large settling ponds, vertical flow ponds, aeration fountains, limestone beds, wetlands, all of the idea of increasing your pH and precipitating the metals on site and keeping them from entering into your watershed. So the Environmental Protection Agency, the EPA, does set limits for both iron and manganese, how much is allowed to be exiting a mine, how much aquatic wildlife is allowed to be exposed to on a continuous basis, as well as drinking water recommendations. So at the Boys Park system, which was that lovely aquamarine blue color we just had on the last slide, this is what the system looks like from Google Maps. So we have a couple of places where the water is coming into it, and it all starts out about a level of 4 parts per million for iron. When it's exiting here into the stream, into the watershed, it's exiting at 1, so it's improved, but it's still above our limits for drinking water and aquatic life. And manganese, it all enters the system about 0.5 or 0.4, so there's not really a lot of remediation going on for manganese in this system, unfortunately. So something we want to look at as biologists and as microbiologists is the influence that microbes that are just naturally in the environment can have on these systems, and the impact specifically on biogeochemical cycling. So we can have microbes that are not so great. We can have biocontamination, where these microbes in this environment here can lead to the loss of an electron and formation of Fe3+, or Mn3+, or Mn4+. Actually, excuse me, the other way here. Fe2+ or Mn2+, which are more likely to be soluble chemicals, and these are more likely to enter into your watershed. So this is bad. It's going to lead to increased contamination and have a negative impact overall. Whereas we can have microbes that are leading to oxidation, so the loss of an electron with those little blue dots in the electron shell, where we have Fe3+, Mn3+, or Mn4+, and these are more likely to form precipitants and fall to the bottom of the ponds and therefore not enter the watershed. So the microbes that are naturally in the system, they could have no impact or high impact or somewhere in between, and we're interested in figuring out which ones they are and how they're doing it. So a little bit of background on biology. This is sort of the central dogma of biology. So all of our cells, even bacterial cells, have DNA, and these DNA will encode like blueprints for genes of function for protein. So in between DNA and protein, we have RNA. Now, bacteria don't have a lot of extra energy, so if they're transcribing something from DNA to RNA, it's most likely going to end up being a protein. But there's this intermediate step in between. And then lastly, we end up at protein, where these are things that have functions. So in particular, proteins such as enzymes can have a specific function and drive a reaction. And in particular, there's enzymes that are called oxidases that drive oxidative reactions, and reductases that drive reductive reactions. So in particular, there's iron oxidases, and these can be part of microbes' electron transport chains, and this is how they generate energy to survive. This is just their version of metabolism. And these can go from Fe2+ to Fe3+, and this can be encoded for in the genes or in the DNA by CC4 or CYCA. Nitrate reductases, where we go from nitrate to nitrite. This can be carried out through the disintegratory nitrate reduction enzyme, which is encoded for NAPA and NAPB genes. In the literature, there is mentions of the linking of these two, so the iron oxidation linking to the nitrate reduction, but this is not fully understood, which is why we have these question marks here. So how do we do this? So what we do is we go out into the field and collect our pond water slurry, and we bring it back into the lab. We will sterilize part of it and keep part of it non-sterile, and at a controlled time, we'll add this non-sterile portion back in and monitor for changes. So whether it's a good change, where we have less contamination, or a bad change. We then will extract DNA from our bacteria that we've isolated out as either positives or negatives impacts, send them for whole genome sequencing, annotate for genes of interest, and see if there's genes that are predicted for metal cycling, such as that cytochrome C4, which cycles iron, and NAPA, which will help cycle nitrate. Once we have identified genes of interest, we can then develop primers which will target that specific gene, confirm that that gene is actually there in the DNA, and then confirm with RNA that it's actually being transcribed, which means it's most likely becoming a protein, which means most likely active or having an impact. And then last, we can look at the level of expression during remediation. So iron remediation. So what we have here is we have this isolate we found, Parabricolaris. This is AB18. And this little guy, if we add him back into sterilized pond water, we get a oxidation of iron. So this is nitrate-dependent iron oxidation. This occurs when we feed it nitrate. Now, this does not happen if we don't add these cells back in. So we can say that this is a biological thing that is occurring, and that this is an iron oxidase. As microbiologists, we tend to characterize our bacteria, so this is just a little selfie of our bacteria here. This is Parabirculdaria. We do these sort of classical microbiology things where we look to see the gram DNA to determine like the membranes that are present, whether it has 1 or 2 layers, capsule and biofilms, just to confirm if they're sticky and they can stick together and form communities. We also look for the pH range, which is not surprising since this came out of an acidic system that it likes the lower pHs. And 16S-RRN is a typical way that microbiologists will identify bacteria by species. We also looked at the genome size and the number of protein coding genes that it can have. And importantly, it does have the genes predicted for NAPA and CC4. So this is a gel. So we use these in the lab to take our primer, amplify a certain piece of DNA that we're interested in, and then we use these gels to image them to confirm whether or not that gene is present. So these are size markers, and this is the gene of interest that we've amplified. So this is NAPA, so this confirms that it's here. This would just be empty if there was not this gene present. Same thing here, we confirm that CC4 is here, and CC4 is here. These are just 2 different primer sets targeting the same gene, which is why we have different sizes, but it does confirm that gene set. So for future work from the iron bioremediation, what we would like to do with the big picture is now that we have a microbe that can impact iron remediation positively, we want to develop bioindicators using those primers we develop to determine how present are these genes in the system, and also determine like how active are they. And we can do that by looking at the RNA or the gene expression, and sort of as a way to monitor system health overall. Another thing too we want to look at is biostimulation. So if we can use these microbes in a way to improve mediation, if we feed them a certain natural carbon source, will this improve? So these are just some sort of things that we need to knock off to be able to achieve those goals. So figure out what carbons it can survive on, figure out what are the optimum temperatures, pH, and oxygen levels for growth, and the impact of this on remediation, and determine what natural carbon sources we can use for in-field. So for the manganese resolubilization or the biocontamination aspects, we have these really cool plates that came from literature. This actually came from a paper, I want to say like the 60s. And this is basically a plate here that has manganese added to it. And it starts out totally brown. When we add back in our pond water on top of it, we see this clearing. So that indicates that something in this massive bacteria right here is capable of reducing manganese. Okay? And we find this actually in all of our ponds. This is just a screen of pond 1. But we have this in all 8 of the ponds at Duquesne. And when we pull out those actual single isolates and we again add them back to sterilized pond water, we see that sterilized pond water, there's not a lot of change by itself, but when we add these isolates, there is an increased amount of manganese that is soluble. So that is microbial driven right here, especially with this gray one here. So for future work for this, we want to continue characterizing this. We want to make sure we can replicate this reproducibly so that we actually know that we're seeing what we think we're seeing, as well as screen for the additional ones. So that was just pond 1 that I pulled out. We have 8 more ponds, and we have 7 more ponds, 8 total. So screen the rest of those isolates, as well as determine how present and abundant they are. So we can try to use those plates to get single colonies to count how many bacteria are present within each pond. We can also develop primers again specifically for those bacteria, and you see how prevalent they are throughout the entire system. So lastly, I just want to say thank you to my advisor, Dr. Nancy Trun. None of this would be possible without her, as well as my committee, who always has great ideas and suggestions. Trun Lab members past and present, we have a whole slew of them. And also the Stoltz Lab, Dr. Cantley, who's been kind enough to run water samples and biology department as a whole, as well as funding sources through the American Society of Microbiology, National Science Foundation, through the Geological Society, and of course, NASLR. So thank you guys. And with that, I would be happy to take any questions. Yeah.
Do you notice any seasonality with this bacterial development?
Yes, that is such a great question. So the question was if there are seasonal impacts for bacteria in the system. So there's actually something a previous PhD student had done before I joined the lab, and she sort of set the groundwork for looking at microbes in these systems. And she actually did a seasonal study. There's a paper out there. I can look it up and share it with you if you're really interested. But we see that there's changes in the level of remediation for seasons and the changes in the level of bacteria and the type of bacteria that are present. saw nothing. Yes, Bishop.
So I know you're still pretty early on in your study, but has there been any preliminary studies on the scalability of this?
So scalability is always a question, right? Especially like when you're taking something from the lab and bringing it back into real life. There are definitely papers that talk about in-field applications of things like this. We're not quite there yet. We want to really make sure we understand what we're doing right before we start poking around the field as scientists, because there can be a lot of red tape and a lot of scary things when we talk about stuff like that. But yeah, that stuff's definitely out there. Thank you.
Could you describe maybe one of the biggest surprises that you had during your research?
Ooh, one of the biggest surprises during research. How long it takes to make things work. That is one of the biggest surprises. I think I spent a whole year just trying to develop a synthetic AMD because of the seasonal changes. Every time we go out to sample, our pond water is slightly different. So then it adds another variable to our experiments as we go. So one of the things I was tasked with was to make this synthetic AMD that replicates 22 chemicals that are naturally found in our ponds. And it took me a whole year to make it, to figure out the chemistry behind it, and then to get it actually to work and replicate the iron oxidation in it. All right, thank you guys so much.
Thank you.